Hello learners, welcome to today's lesson. Today we'll be looking at biology, paper 2 theory 2021 internal. This is your presenter, Casey. Question number 1. Figure 1.1 and figure 1.2 show freshly cut potato chips suspended in solution E and F after the beginning of an experiment and after 2 hours respectively. So here we can see the beginning of the experiment in figure 1.1, and after 2 hours we can see what's happening here. Question number 1A. Explain the difference in appearance of potato chip in solution E and F after 2 hours. So we can see after 2 hours that potato chip in solution E shrank, and after 2 hours potato chip in solution F gained water and became big. So here is our answer. The potato chip in solution E loses water by osmosis, while the potato chip in solution F gains water by osmosis. That's the answer. Question 1-2. Based on the apparatus of the potato chips in solution E after 2 hours, what term can be used to describe solution E? Based on the appearance of the potato chip in solution E after 2 hours, what term could be used to describe solution E? So solution E is a hypotonic solution. B1. What would be the state of the cell in the potato chip in solution F after 2 hours? So after 2 hours, what's the state of the cell? So the cells will be turgid because they have gained water. B2. State the importance of the state of cell in B1 above the plants. So state the importance of the cells in B1 above the plant. So the importance of turgidity. So it helps support the plants. C. Explain the meaning of the term isotonic solution. This is a solution which has the equal amount of salt solution as the inside of the cell. So these were the answers for question 1. Question number 2. Table 2 shows various food tests carried out by a learner. So this is the food test. In the nutrients, we have the tests, the observation, and the conclusion. Complete the table by filling in the spaces. So number 1, we have starch. So starch, what is the test for starch? What do we test with starch? And that is iodine solution. We use iodine solution to test for starch. 2, we have immersion. So what are we testing what are we testing for immersion? So here we have the fats. So when we observe white precipitate, then we are saying that fats are present. And here we have the protein test for burette. Proteins are present. So the color changes from blue to purple. So that's 4 marks. That's 4 marks. Yeah. Let's carry on. A2. Which nutrient in the table would cause kwashiorkor if lacking in the human body? Which nutrient in the table would cause kwashiorkor if lacking in the human body? That's proteins. A2. Name a nutrient not in the table which helps to prevent constipation. Name a nutrient not in the table which helps to prevent constipation. That's roughage. State 2 deficient diseases in plants. So we have 1, yellowing of the leaves, 2, stagnant growth. Question number 3. Figure 3 shows a longitudinal section through a root tip. So this is our root tip. We have vascular bundle, G, H, I, and J. These are the regions. So question A. Identify the parts labeled G and J. We are talking about these parts. And G is the root hair. Those are root hair cells. And J is the root cap. So those are the answers. B. Which labeled part in figure 3 is responsible for generating new cells? And the region is region I. Region I is responsible for generating new cells. So yeah, region I is the region of cell division, and it's responsible for generating new cells. C. Region H is responsible for cell differentiation. Apart from vascular bundles, state 2 other cell types differentiated by region H. So we're talking about region H, and what cells are differentiated in region H. That's the sclerenchyma and the parenchyma tissues. So those were answers for C1. C2. State 2 types of cells found in vascular bundle. So the cells found in the vascular bundle are xylem and phloem. Which named type of cell in C2 allows movement of substances upwards and downwards depending on the need of the plant? C3. Which named type of cell in C2 allows movement of substance upwards and downwards depending on the need of the plant? So this is the phloem. The phloem can take the food either upwards or downwards, but the xylem only takes the food upward, only takes the mineral salts and water upwards. So here our answer is phloem. D. Suggest one merismatic structure not in the diagram responsible for bringing about secondary growth in dichotomous plants. So here, that's the cambium. Question number 4. Figure 4 shows the structure of a nephron. So this is a nephron. This is just a part of, it's found in the kidney. There are a lot of them in the kidney. So this is just one nephron. A. Identify the parts labeled K and L. So we are identifying K, and this is K. We are also identifying L. So here K, this is the afferent end. L is the Bowman's capsule. B1. Name two substances present in the fluid in K but absent from the fluid M. So we are talking about when the fluid has just entered the kidney in the afferent end and has been filtered out in the glomerulus. So we are talking about after filtration, after ultrafiltration, what remains in the fluid. So what will remain in fluid M? So we have one, blood cells are not present in M. Since they are too large, they are not present. And plasma proteins are also not present. They are not filtered out. So once they are not filtered out, they continue their way, as we see as we see the arrow point in that direction, but the rest they continue in tube M. So these were the answers for B1. B2. Give two reasons for your answer in B1 above. Answer 1. They are too large to be filtered out. So these are too large to be filtered out in the glomerulus. 2. The walls of the glomerulus are too thin. Those are the reasons why these are not present in fluid M. C. State one common disorder associated with the kidney. Here our answer is kidney failure. Explain how the disorder in C1 can be treated. So kidney failure can be treated by kidney transplant or using a dialysis machine. These were the questions. These were the answers for question 4. Let's move on. Question number 5. Figure 5 shows 4 animal cells at different stages of mitotic division. Figure 4 shows 4 animal cells at different stages of mitotic division. So we can see here there are 4 animal cells and each at a different stage of mitotic division. So this is mitosis. So what's happening is mitosis division. A. Name the structures labeled N and O. N and O. We can see N is just pointing there. So N will be the nuclear membrane and O will be chromatids, as we can see here where N is pointing and O is pointing. B. At which stage of mitotic division are cells 1 and 3? At which stage of mitotic division are cells 1 and 3? So we are looking at cell 1 and 3. So cell 1 is at anaphase, cell 3 is at metaphase. Let's carry on. C. A husband and wife were heterozygous for blood group A and B respectively. With the help of a genetic diagram, show the possible blood groups of their children. So here, make sure you follow all the steps as I will be doing for you to score all the marks at this stage. So first, we have the parental phenotype, that's blood group A and B heterozygous. We have the parental genotype, AO and BO. Make sure you put them under circles. Make sure you enclose them in circles. So here we will come at the gametes. The gametes are A and O, and B and O, and also make sure you also put them in circles. Don't leave them without circles. So now here you have to show the gametes where they are coming from with the arrows. And again, make sure your lines or arrows are touching the circles. Don't just put them anyhow. Make sure they are touching the circles. So here now we'll come at offspring genotype. So after this is now the fertilization will take place here, and this will be our offspring genotype. Hope you have followed on this step. So here we have the fertilization taking place. Just in case you have missed, let me just go back slowly so that you can see where the lines are pointing. So A is pointing with B. We have one child having AB, and again A will be pointing with O, another one having A heterozygous A, and O will be pointing with B. That will be B, BO. Make sure to always start with the B, not with the O. And the other one will be blood group O. Make sure you follow these steps as I have done it for you to score all the marks. So you even have to write the offspring phenotype, that's blood group A, B, blood group A, blood group B, and blood group O. So those were the answers for question number 5. Now we have come to section B, essay questions. So the instructions are: answer any 3 questions from this section. All questions must be complete sentences and paragraphs. Write your answer in a separate answer booklet. So remember that your answers must be in complete sentences and paragraphs, not in point form. You have to join your points using words, or you have to use commas or full stop. They should not be in point form but should be in paragraphs. So here you only have to choose 3 questions, but for the sake of revision we are going to do all of them. So let's get right into it. Question number 6. Describe the movement of organic solutes in the phloem tissue. B. Outline the process of measuring the rate of transpiration using a potometer. C. Explain the factors that affect the rate of transpiration. Question 6 answers. A. Organic solutes move through the phloem by the process of translocation. Translocation is the movement of manufactured food from the source to the sink site. The source is the point of origin and the sink site is the point of storage or usage. The organic solutes mainly consist of sucrose and amino acids dissolved in water. The leaf in most cases are the source. B. To prepare a potometer, first obtain a potometer, fill a beaker with water, put the delivering tube end of the potometer into a beaker of water, make the whole setup completely filled with water, put the young leafy plant in a basin of water, cut the stem while the plant is in the water, quickly transfer the cut shoot into the open end of the potometer and fix it in position with the end of the cork and attach a rule onto the delivering tube of the potometer. Lift up the delivering tube to allow air bubbles. Expose your set to bright sunlight outside your laboratory and set the stopwatch when the air bubbles reaches the 0 mark of the rule. So that's how you set up a potometer. C. The effect of transpiration are temperature, humidity, light intensity, and wind. The factors that affect transpiration are temperature, humidity, light intensity, and wind. The higher the temperature, the faster the rate of transpiration. The higher the humidity, the slower the rate of transpiration. The higher the light intensity, the faster the rate of transpiration. And the faster the wind speed, the faster the rate of transpiration. So these were the answers for question number 6. Question number 7. A. Describe the internal structure of the heart. B. Describe the factors that reduce immunity to pathogenic diseases. C. Explain the importance of immunization. A. The heart is a muscular organ located in the chest between the lungs. It is made up of muscles known as cardiac muscles which contract and relax without getting fatigued. The heart is divided in 4 chambers, namely the right and left atrial and the right and left ventricles. The chambers are separated by the septum. The heart also contains valves known as tricuspid and bicuspid valves and semi-lunar valves. B. Factors that reduce immunity include poor diet, development of resistant strain of pathogenic Factors that reduce immunity include poor diet, development of resistant strains of pathogens, repeated invasion by pathogens, intake of immunodepressant drugs, and infection by HIV that destroys the lymphocytes. C. Immunity is important for prevention of diseases such as measles. Passive immunization raises the number of antibodies in the bloodstream, and active immunity, the body makes memory cells that produce antibodies more rapidly the next time there is a similar infection, hence keeping person healthy. Question number 8. Using named examples, describe animal and wind dispersal of seed and fruits. B. Explain the importance of fruit and the seed dispersal. 12 marks in total. A. Animal dispersal. Dispersed seed have the following characteristics: presence of hooks to cling to the fur of animals, e.g. blackjack. The hooks irritate the animal, causing it to shake the seed off some distance away from the parent plant. They are juicy to attract and reward animals, e.g. mangoes. They are brightly colored to attract animals to fruits, especially when ripe, e.g. oranges. Seeds have hard testers that are resistant to digestive enzymes found in the gut of animals, e.g. guavas, and they have scented fruits to attract animals, e.g. oranges. Wind dispersed seeds have the following characteristics: parachute of hair formed from sepals after fertilization, causing the fruit to float, thereby delaying landing and encouraging dispersal. An example of seed is the dityrion seed. Wing-like structures that cause the fruit to float, thereby delaying landing and encouraging dispersal. An example of a seed that has wing-like structures is the sycamore seed. They are also numerous and are not heavy, so that they can be easily carried by the wind. B. The importance of seed dispersal is that it reduces competition for light, space, and nutrients. It allows plants to grow in new areas, and it reduces the chances of extinction, and it also allows propagation of some seeds because they only germinate after passing through the digestive system of an animal. Question number 9. Describe the function of the following: 1. Thorexin. 2. Glucagon. B. Explain the causes of short sight and how it can be corrected. C. Suggest ways by which deafness could be prevented in humans. Thyroxine. Thyroxine is produced by the thyroid gland, and its main function is to control the basal metabolic rate of the body, stimulate respiration of glucose and fats, and controls the growth and differentiation of cells. Glucagon. Glucagon is produced in the pancreas and is responsible for converting glycogen to glucose in the liver and the muscles when sugar levels drop in the blood. B. Short sight is a condition where a person is able to clearly see objects at a short distance but cannot see clearly objects far as they appear blurred. It is caused by having a permanent thick lens and abnormal long eyeball, which makes the image form before the retina. It can be corrected by wearing a concave lens. Deafness is where hearing is partially or totally diminished. It is brought about by accumulation of wax in the ear, blocking the external auditory canal, infection to the inner ear, and physical damage to the eardrum by placing sharp and hard objects in the ear. It can be prevented by cleaning the ear regularly with soft objects, avoiding loud and noisy places, and by treating infection with care and proper medicines. So these were the answers for question 9. Question number 10. Explain the meaning of the following ecological terms. 1. Habitat. 2. Community. 3. Niche. B. Using named examples, explain what a food chain is. Using named examples, explain what a food chain is. B. Explain how energy flows through the food chain. Habitat. A place where an organism lives. Examples of habitat are aquatic habitats in water and terrestrial habitats found on land. So a habitat is a place where organism lives. Community. A group of population found in the same area and interact with each other. A group of population found in the same area and interact with each other. Niche. The specific role an organism plays in an ecosystem. The specific role played by an organism in an ecosystem. B. A food chain is a sequence of feeding relations that begin with the producers and involves at least 3 organisms. The feeding level of an organism in a food chain is called the trophic level. In every food chain, the producers occupy the first trophic level and the primary consumers occupy the second trophic level. The secondary consumers occupy the third trophic level, and so on. An example of a food chain is given below. So here is an example of a food chain. Cabbage grows. When the cabbage grows, it is eaten by the rabbit, and the rabbit is later on eaten by a man, and the man is eaten by a lion. So this is an example of a food chain. The principal source of energy for an ecosystem is the sun. Energy from the sun is called solar energy. Solar energy is captured by green plants during the process of photosynthesis and converted to chemical energy, which living organisms are able to use. Because of their capacity to produce food for other living organisms in an ecological system, in an ecosystem, plants are called producers. Animals that feed on plants are called primary consumers or herbivores. Those that feed on primary consumers are called secondary consumers, and those that feed on secondary consumers are called tertiary consumers. Food chains can rarely go beyond the fourth trophic level. This is because only 10% of the energy present in one trophic level is passed to the next. 90% of the energy is lost through respiration, ingestion, and excretion. By the time a food chain reaches the fourth trophic level, there is very little energy available for any higher trophic level. This is more energy gained by feeding on vegetation than feeding on meat. So these were the answers for question number 10. Make sure you read them through so that you can understand them thoroughly. We have come to the end of this program today. If you have found this video helpful, make sure you subscribe and like. And if you are interested in this revision past paper, make sure you text me or call me in the number in the description. Can't wait to see you in the next lesson. Goodbye.